Can you hear me? I hope this is okay. Can you hear me in the back? Whoever's further away, can you hear me? So let me just play it once more. Right. So this is how you imagine a lipid graft. Here you have a surface view of a cell with the head groups of the lipids facing the aqueous phase. And this is the idea that the membrane would have these patches of regions that are enriched in cholesterol. And this here indicates a GPI anchored protein that you can see here that is linked in a different way to the bilayer, and some transmembrane proteins. So this is something that may come up in many things that you hear about signaling later on. So what we want to do today is just take one step down into the cell, right? So we want now to start talking about these membrane organelles that you have below the plasma membrane. So the cell has a large number of these very complicated networks of membranes. And what you can see here that the most important ones are indicated, that I'm sure all of you already heard about. Right, you have the very dynamic system of endosomes that keep coming into the cell, but into the plasma membrane. And then on the other direction, you have secretory vesicles coming out of the endoplasmic reticulum and the Golgi complex, fusing with the plasma membrane. And you have the mitochondria, right, which we already looked at. They are believed to have been acquired by endosymbiosis in the early evolution of cells. And you have several other membrane-bound organelles that we're going to be discussing as we go along. So what you want to think is, first, what are the organelles that occupy more volume in the cell? So this is my first question in the class, right? So you have this table here. So the relative volumes occupied by these intracellular compartments, right? So what is listed is the cytosol, mitochondria, the endoplasmic reticulum, which can be divided into rough and the smooth PR, the nucleus, the peroxisomes, the lysosomes, and the endosomes. So who wants to tell me which one of these categories here you think represents the largest percentage of the cell? The cytosomes. So everybody got that right. So the interesting question is the second largest. What is the second largest?
Mitochondria. Mitochondria.
You said something? I don't know where it came from.
Mitochondria.
Which one you said?
Mitochondria.
Mitochondria, good. You don't get this one. Mitochondria are, and why is it that mitochondria occupy so much space in terms of volume? The reason when we get to the amount of membranes, right? Because they have those internal membranes that are extremely convoluted, so the volume of the mitochondria is actually large. So here's the answer, right? Oops. There it goes. So cytosol is the highest percentage, up to 50%. Then you have mitochondria, and then after that comes the endoplasmic reticulum. But this, as we're going to see, this can vary depending on the cell type. So let's take a look at the ER. And there's one animation here that really gives a good idea of what is the endoplasmic reticulum. Right? It's called the reticulum because it really forms this. I don't know why we're not having sound. I just decided to shift to the computer, and now we have a technical issue. Bear with me, I'm going to shift my iPad because there's no sound on the VGA. So we're gonna be thinking now about the endoplasmic reticulum.
Is a highly dynamic network of tubules that dissolve into periodic cells, like a spider's web.
Still very low, huh?
The network is growing
This classroom.
with some connections being broken, while new ones are being formed. Motor proteins moving along microtubules can pull out sections of endoplasmic reticulum membranes to form extended tubules that then fuse to form a network.
Right, so this of course is not the real ER in a cell, so this was actually an in vitro experiment that scientists have learned now to really isolate the ER and spread it on glass, and then show that these tubules are actually controlled by these motor molecules that you're going to be hearing about in the cytoskeleton lecture. So what is really important in terms of the endoplasmic reticulum is its role, right, in membrane traffic. So this is a term that we use a lot in cell biology because there's no such thing as static compartments bounded by membranes in cells. They are in constant flux. There's membrane budding and fusing, and it's really a fascinating process how this equilibrium is maintained. So who wants to tell me? So this means if you now, instead of thinking of volume, you think of the amount of membrane in cells. So we have two types of cells here. Let's think first of the hepatocyte, which would be a cell that does a lot of protein synthesis. Which membrane compartment of these listed here you think is more abundant with these cells? Smooth ER. Everybody agrees that would be the smooth ER. And why?
Because the liver is responsible for cleaning up toxins, and the smooth ER is responsible for smoothing.
But I just mentioned that another thing that the hepatocyte does is a lot of proteins. What is
Rough ER
Yeah.
then. It's that part. Rough ER then.
You're right that the hepatocyte is a complicated cell that does a lot, right? But the numbers here, it would be still more in the rough ER, right? Because I think the protein synthesis of the smooth ER beats the other functions, but you see that it has a very upregulated ER. But the second compartment is still the mitochondria, right? So, and you see here that the contribution of the inner membranes of mitochondria, it's really very high. So that explains really why mitochondria look very compact, but they actually are second place in terms of abundance of membrane. So what if we think, so we already got on this thing, this means that the cells do different things. So what if we think now of a cell from the pancreas that does a lot of secretion? So it puts a lot of protein out of the cell. So which compartment you think would be more abundant in this cell? Anybody?
I guess the Golgi.
You think the Golgi would be
Yeah.
more, the Golgi would be more developed in this cell? So what do you think?
I would say rough ER because we need more proteins.
Right, so this is an interesting thing to think, right, of cell function. This means it's true that the Golgi is an essential station to put proteins out, but if you don't make the proteins first, so what is the compartment that really reacts to this need? So you see here, it's really dramatic, right? This means, so pancreatic cells have a lot of rough ER. And I think I have an electron micrograph here. So this is what it looks like, right? So I think you all already saw this. So these black dots are the ribosomes attached to the cistern of the rough ER. Yes.
So why did it have a lot of rough ER?
Yes, so was this discussion that it is true, right? This means, let me put back, I move very fast. So this means if you look at the Golgi is still elevated, right? So one of the answers we got is that the Golgi would be higher. It's true. But for the proteins to be sent to the Golgi, they first have to be made. So you need a lot of rough ER, which is where the ribosomes are, to produce proteins that are then going to traffic out of the cell. So this is the typical pattern, right? And this is the technique that I mentioned before, right? The transmission EM, in which the cells are sliced very thin, and then they are treated with metals to give contrast. And each ribosome appears as a dot. And this really comes way back from those first studies of George Palade, that people learned at that time that the rough ER really was a very dynamic organelle, that depending on the function of the cell, you saw a huge expansion of this compartment. So this is the way to think about this in 3 dimensions, right? Which is not very easy to imagine this. So, but this is how another electron microscopy technique, which is called tomography. So it's possible to do that slicing process, going down into the cell and then reconstructing this the computer. You had a question? So this is what scientists arrived at, that the rough ER tends to be like these flat pancakes. So the area actually is larger, that you have a lot of space to attach the ribosomes. And then they converge in this much more tubular and irregular area, which is the smooth ER, which looks more irregular. And I think I have an electron micrograph here. So this would be a section of a cell that has a lot of smooth ER, right? So it's not that easy to identify smooth ER compared to the rough ER, because first you don't see the ribosomes, and second you see that some of these tubules are in cross-section and some are on longitudinal section, so it appears very messy and irregular. But if you imagine this then connected to the more flat cisterna, which are the ones that hold on the ribosomes. So this is what we want to do this, right? I know all of you or most of you already had this in cell biology, but what we want to do in this course is to try to imagine this in 3 dimensions and really correlate this with function, right? So one of the functions of the ER that is not that well known is the synthesis of lipids, right? So we were talking a lot in the previous class about the several classes of lipids, right? There was a little bit of confusion with the question on the video, but I think it was just the way the question was formulated, right? Gito Mayor, in his video, he mentioned that there are more than 500 types of lipids. So some of you answered that there were 500 types of lipids, and some of you just says, well, there are 5 classes or there's 3 classes, right? And so it just depends how you classify them. What he meant is that he was talking about all the variations that can happen on the head group and also on the degree of saturation, right? So if you think of different molecular species, there's more than 500. But they fall into these categories, right? In which they are the phospholipids, like we mentioned before, they're the most abundant ones, that have phosphate in their head groups. So this here is indicating this machinery that sits on the cytosolic side, right? Again, think of topology. So this would be the bilayer of the ER, in which you have here on the top the cytosolic side, and then down here would be the lumen of the ER. So there is a really complex and very elaborate machinery that sits on the cytosolic side that has several enzymes that sequentially they build up these lipids. So they start with these hydrophobic tails that are inserted on the membrane, and then these enzymes gradually add the head group. So this happens on the outside. And we already discussed that in some cases, I'm going to show you more examples, that after all this is assembled, it has to flip to the inside. So there are enzymes also that promote this flipping. So this is an example of how phosphatidylcholine would be built first, forming phosphatidic acid, then diacylglycerol, and finally phosphatidylcholine with the addition of the choline head group. So we should think then of the ER not only as this factory of proteins, right, where the ribosomes are ready to attach proteins and insert proteins into membranes, but also these two other functions, that they are the place where lipids are synthesized in cells on the cytosolic phase of these tertiary. And what I mentioned in the last class also, that the ER has this very important function of being a store of calcium. So these red dots in this slide, they represent calcium, which is very low in the cytoplasm, right? So anybody remembers how many times lower is the cytosolic calcium compared to the outside? Which order of magnitude? Second? 10,000. So maybe it's easier to think in nanomolar versus millimolar, right? Millimolar outside, nanomolar outside. And inside the system of the ER is again millimolar. So this is a very important signaling mechanism, and the ER plays an important role. So we went through three functions of the ER already, right? Protein synthesis, lipid synthesis, and storage of calcium for signal. So let's think now of the problems that cells have to solve. And one important problem is, right, so at the time when we talk about protein synthesis, everybody imagines the ribosome and messenger RNA being copied and translated, but there's a big difference between making a protein that is going to live in the cytoplasm to a protein that has to insert on the membrane. So this is something that scientists were interested in a lot before they really knew the structure of cells. And this was a really very important achievement to understand how newly synthesized proteins are inserted into the ER. Right? So let's listen to this animation, and then I'll tell you more about it.
The endoplasmic reticulum, or ER, is the most extensive membrane system in eukaryotic cells. Proteins transported to the Golgi apparatus, endosomes, lysosomes, and the cell surface all must first enter the ER from the cytosol. As an mRNA molecule is translated into a protein, many ribosomes bind to it, forming a polyribosome. There are two separate populations of polyribosomes in the cytosol that share the same pool of ribosomal subunits. Free ribosomes are unattached to any membrane. Membrane-bound ribosomes become riveted to the ER membrane and translate proteins that are translocated into the ER. These membrane-bound ribosomes coat the surface of the ER, creating regions called rough endoplasmic reticulum. Two kinds of proteins are moved from the cytosol to the ER. Water-soluble proteins completely cross the ER membrane and are released into the lumen, while transmembrane proteins only partially cross the ER and become embedded in the membrane. All these proteins are directed to the ER by a signal sequence of small hydrophobic amino acids. The signal sequence is guided to the ER membrane with a signal recognition particle, or SRP, which binds the ER signal sequence in the new protein as it emerges from the ribosome. Protein synthesis then slows down until the SRP ribosome complex binds to an SRP receptor in the ER membrane. The SRP is then released, passing the ribosome to a protein translocation channel in the ER membrane. Thus, the SRP and SRP receptor function as molecular matchmakers, connecting ribosomes that are synthesizing proteins containing ER signal sequences to available ER translocation channels. In addition to directing proteins to the ER, the signal sequence functions to open the translocation channel. The signal peptide remains bound to the channel, while the rest of the protein chain is threaded through the membrane as a large loop. Once the protein has passed through the membrane, it is released into the ER lumen, after the signal sequence has been cleaved off by a signal peptidase located on the luminal side of the ER membrane. The signal peptide is then released from the translocation channel into the membrane and rapidly degraded. It is thought that a protein serving as a plug then binds from the ER lumen to close the inactive channel. But not all proteins that enter the ER are released into the ER lumen. Some remain embedded in the ER membrane as transmembrane proteins. For clarity's sake, the membrane-bound ribosome will be omitted to illustrate the translocation of transmembrane proteins into the ER membrane. In the simplest case, that of a transmembrane protein with a single membrane-spanning segment, The N-terminal signal sequence initiates translocation, just as for a soluble protein, but the transfer process is halted by an additional sequence of hydrophobic amino acids, a stop transfer sequence, further in the polypeptide chain. The stop transfer sequence is released laterally from the translocation channel and drifts into the plane of the lipid bilayer, where it forms a membrane-spanning segment that anchors the protein in the membrane. As a result, the translocated protein ends up as a transmembrane protein, inserted in the membrane with a defined orientation. In some transmembrane proteins, an internal signal sequence is used to start the protein transfer, which continues until a stop transfer sequence is reached. The two hydrophobic sequences are then released into the bilayer, where they remain anchored. In complex multipass proteins, in which many hydrophobic regions span the bilayer, additional pairs of stop and start sequences come into play. One sequence reinitiates translocation further down the polypeptide chain, and the other stops translocation and causes polypeptide release, and so on, for subsequent starts and stops. Thus, multipass membrane proteins are stitched into the lipid bilayer as they are being synthesized by a mechanism resembling a sewing machine.
Okay, so this is how these transporters that we looked in the previous class are really synthesized. So they are, it's coupled to translation, this insertion in the membrane. And this was a major breakthrough, which I think is considered the first really Nobel Prize of modern cell biology, in which Gunther Blobel at Rockefeller University really figured out, was really an inspired work that followed, right, also at Rockefeller. So Rockefeller University really was the cradle of modern cell biologists, because it was there also that Claude Leduc and Palladi did the first electron micrographs and started realizing that cells had all these membrane compartments. But Gunther Blobel really went further, and he did biochemistry, and he did some really elegant experiments, and he is known for what is called the signal hypothesis, right? That it is a sequence on the protein that really determines how it is translocated or anchored in the ER. So the experiments that his lab did were really elegant, because first they did some of the first fractionations that were done in biology. So they did this preparation that up to this day we still call this microsomes. So you basically get a cell that has a lot of ER and homogenize this in the lab. You end up with these vesicles, because the membranes break and then reseal, right? So this means this is an aqueous solution. And you end up with these vesicles that are like this, with, you see here that the ribosomes are facing out. So you have these really useful, more or less homogeneous vesicles that are made of rough ER. And of course, there are more details into this. You don't just homogenize, you do also density centrifugation. And doing this centrifugation in these gradients, it's possible to isolate fractions that are more dense because they have the ribosomes. So these were the ribosomes that were used for the experiments. And one of the important insights that happened here is that when they mixed, right, messenger RNA with these purified ribosomes in a cell-free system, they found that the protein that they obtained was shorter in the presence of microsomes and was found inside the lumen of the vesicle. So they showed that translocation occurred, and that during translocation these proteins were losing a piece, right? Which was really the beginning of the discovery of the signal sequence. And what they used to determine that it was inside the vesicle is what we call protease protection. It's clear to everybody how this works. How can you use a protease to show that something is on the other side of the membrane? It has been translocated inside the vesicles. Imagine that they have this mixture in a test tube, and then they know a protein was made, but they don't know if the protein is inside or is outside. Exactly. So the key property here of membranes, right, is that these membranes would be impermeable to a large protease, which is also a protein. So the protease on the outside would degrade all the protein. So they only ended up, so they were only interested in what was protected. And they saw that these rough microsomes were capable of promoting the protein to enter the vesicles, while if they use the smooth microsomes, which are the part of the yarn not containing the ribosomes, this didn't happen. So this is what really was the beginning of the understanding now that there are really two cycles of ribosomes in cells. So you have proteins being synthesized on free ribosomes, right? This happens in the cytosol. And there are a lot of proteins that never go into membranes or across membranes. So these proteins are synthesized on the free ribosomes. And then you have the membrane-bound ribosome cycle that is really this fascinating machinery that took a long time to figure out. But this machinery really sequentially is what you just saw in this animation, that the signal recognition particle and the translocator pore really function sequentially to really get these proteins inserted into the membrane. And what's really remarkable of this translocation pore is that you saw that a very important property is that the portions of proteins that are hydrophobic and they are inserted in that channel, they are able to diffuse laterally, right? So this is not a channel that blocks diffusion. It's very important that the lipid portions actually can diffuse out. So this is how the proteins get inserted on the membrane and the ribosomes can continue their cycle. So the two major components that are pretty well understood now, right, is the signal recognition particle, which this really was a major breakthrough because it really explained not only how, so this region of the protein recognizes the signal sequence on the protein, and then there is another domain that pauses translation to really allow this very smooth process of sequentially coupling the translation to membrane insertion. And you can see in this diagram here how the signal recognition particle attaches to ribosomes. So this is all understood now from crystallography studies. Again, right, proteins purified in large amounts, and then you get X-ray diffraction, and this is how the SRP sits on a ribosome. You have the two units of the ribosome here, and so this is something that is getting better understood every day. There's a lot of very active work on ribosome structure. And what is interesting is that this is not the only system, right? Because there are many types of proteins in cells, right? We already talked a little bit about the GPI-anchored proteins. So the GPI-anchored proteins is something completely different because that lipid sugar moiety that serves as the anchor is pre-synthesized on the membrane, and then the protein is attached to it. So it doesn't have to use this signal recognition particle or the translocation pore to insert on the membrane. So another example of an exception of that system is what we call the tail-anchored proteins. So what a tail-anchored protein would be, and this is pretty common, proteins that have a hydrophobic sequence on their C-terminus. So, and there's very few, these sequences are short, so they are sufficient to span the membrane, but they're not long enough to really be recognized by the signal recognition particle when it goes through the tunnel of the ribosome and the translocome. So the way cells solved this was to really come up with additional proteins. So one is the pre-targeting complex. So there is a protein in the cytoplasm that first binds to this hydrophobic region at the end of the protein. And then there's a second protein that is also in the cytosol normally, which is called the GIP3 ATPase. So the pre-targeting complex takes the protein to this complex that is an ATPase, And then when they are complex, this is targeted to receptors. So this is a thing that is going to come up all the time, right? This means that a lot of cytosolic proteins participate in membrane attachment or signaling because they have receptors that are sitting on the membrane. The receptor by itself doesn't do anything, but when this cytosolic protein carrying a recently synthesized protein binds to it, then this protein promotes the insertion of just that hydrophobic tail into the membrane. Right? So this is a different mechanism that also solves the problem of how a protein that is being synthesized on the cytosolic side ends up on the membrane. So I mentioned to you before, right? This means this shows up everywhere, the central dogma of molecular biology, which this is the dogma that holds for every cell that we know, that the genetic material is DNA, and this is copied into RNA, and the RNA is translated into protein. So cell biologists really like to say that there's more to this dogma, right? So this means you have the molecular cell biology central dogma, in which if you just make protein, this is still useless for a cell because what is critical is to get it to the right place. So these two additional steps of the folding of the protein right after it's synthesized, and then how this is targeted to the place in the cell where it's going to do its work. So a lot of the interest of cell biologists has been over the last few decades to understand how these steps work. And I'm going to give you a few examples of what is important here. So we talked, right, about carbohydrates, how sugars attached to proteins and attached to lipids are usually on the outside of cells. So it is again that topology concept. So if we think now how the sugar is attached to proteins, what is the part of the yard that is equivalent to the outside world? You have the cytosolic side and you have the luminous side. Right? Everybody has this clear? Right? Topology is very important. So what is this is here indicating is that the machinery to attach sugars to proteins, right? This is happening on the lumen of the yard. So what you have is this precursor, which is a lipid link to a common carbohydrate, which is, this is the classical N-type glycosylation that happens in many of the glycoproteins. So you have acetylglucosamine, mannose, and glucose, and this anchored to a dolicol moiety on the membrane, which is a very strong hydrophobic group. This works together with a very important enzyme that has multiple passes through the membrane, and it's sitting there on the membrane of the yarn, right? It was synthesized like the way we just saw. And this enzyme has the capacity of promoting the binding covalent of an asparagine on the protein to this precursor carbohydrate. So transfers it to the protein. This is the beginning of glycosylation. So this is happening on the lumen of the yard, and we're going to see when we look at the Golgi that then these sugars keep being processed. So this means they are trimmed, they are modified, and this is a very important process, and this is being prepared to be exposed to the outside world. So sugars are usually looking at the outside of cells, but this is where they start, inside the yard. So one important sequence that occurs, which is an interesting example of this flipping that has to occur, right? So cells had to solve all these complicated problems in which I just told you, this is kind of apparently contradictory, that I told you that this sugar is attached to proteins on the luminous side of the yard. But at the same time, we know that the synthesis of these lipids, which are the precursors, right? You first have to make this dolicol lipid, and there are these high energy nucleotides that really donate energy and its phosphorylation events that are creating these increasingly high energy bonds that at some point, when some sugars are already attached on the cytosolic side, this whole thing flips in. So this flips into the ER, and then it's inside the ER that the other enzymes are there to finish this extension of this precursor oligosaccharide that is going to be attached to proteins. So the cells really solve the problem of basically splitting the work, that some of the enzymes involved are on the cytosolic side and the final enzymes are on the outside. And this also allows the cell to control the process better, right? Because these last steps are only done when you have proteins waiting, because they were just translocated. While in the cytosol, these precursors are usually more abundant than the information. So what is fascinating in this process of the sugars is because the importance of sugars, right? Sometimes it seems like something boring, that this means that sugars are full of these branched proteins, are full of these highly branched sugar structures. But they play a really critical role because they work as tags for the cell to know if a protein is folded properly, right? So you saw that one of the important steps in the central dogma of cell biology is to fold properly the protein. So how does a cell know that a protein is folded properly and it can then be sent to its destination, right? So this is just one example that the membrane of the ER has these proteins. Only one is indicated here, but there are several. Over here it's calnexin, so it's a membrane-anchored ER protein that recognizes carbohydrates. So everybody knows what's the other name that we use a lot for proteins that recognize sugar? Lectins, right? So calnexin is a lectin. It's an ER resident lectin. And the way it works is that, as I told you, right, this means say that a protein was recently synthesized and just received in its asparagine this oligosaccharide group. What happens is that glucosidases in the cytosol then start trimming these glucoses. So when they get to the last glucose, this is a signal to bind to calnexin. So calnexin can only see a terminal glucose. If there's 3, it doesn't see it. So this means that the protein has to spend some time, right? Remember, see here that this slide is again inverted. You always have to look which side is the lumen, which side is the cytosol. So in the lumen of the ER, there are these glucosidases that start trimming that. When they reach this point, calnexin captures it. And by being captured by calnexin, then this really allows the protein to start folding. The calnexin is a chaperone that helps the protein to start assuming its right conformation. And what's fascinating is that there is a system to really check if this works, which the critical component here is the glucosyltransferase. So it is an enzyme inside the lumen of the ER that this enzyme can only recognize a protein chain that is incompletely folded. So if it binds back to glucosyltransferase, the glucosyltransferase is going to start again. So it's going to again attach a precursor oligosaccharide to it, and it's going to send it back to calnexin. So it's like an assembly line, right? And this means that the glucosyltransferase is basically the quality control. So if this protein is not looking right, it looks like it needs a sugar again, it just takes it over and does it again. And the proteins that are properly folded with its carbohydrate really in the right place are allowed to travel outside of the ER. And we're going to talk a lot about how then these proteins that are in the lumen of the ER are then sent out. But this is the very important discovery also that it was more recent, which is the quality control in the ER, right? That it's basically sugar addition to proteins plays a major role. Oops, so I'm skipping one. And this is what is called the unfolded protein response, right? I think many of you must have heard this. So an important component of this process is the proteasome, at the end. So in the cytoplasm, you have this really large kind of barrel-like protease complex that degrades unwanted proteins. So say that several of those cycles that I just showed you went on, the proteins are still incorrectly folded. There's a point in which the cell gives up. And it's very important to clear this because a lot of, for example, neurodegeneration diseases, there are several diseases that are really caused by aggregates forming, because if a protein is not properly folded, it tends to aggregate. So the cell has this very important mechanism, which is to prevent the accumulation of unfolded proteins. So what the cell does is to basically send these proteins to a specific translocator. So there are other chaperones, disulfide isomerase and other likely proteins that recognize the sugar part. And this sends the unfolded protein to these translocators. So they are membrane proteins on the ER that their job is to let the bad proteins come out. And then in the cytosol, you have glucosidases that trim the sugar out. There are ATPases that help with adding these tags, right? Ubiquitin ligases. These chains of polyubiquitin are known signals for the proteasome. When cells see proteins tagged like this, it knows that it should break it up. So this is just sent to the proteasome, and it breaks it down, and then the amino acids can be reused. So this is an important quality control that cells have that is called the unfolded protein response. So let me just summarize for you, because I'd like at the end of the class give a little written exercise for you people, so we don't have that much more. In this part about the ER, the important concept here is that the signal, right? I mentioned to you the big discovery of multi-global, which is still known as the signal hypothesis, is that a signal in the amino acid sequence of proteins determines how they are transported across membranes. And usually it's a hydrophobic signal because it has to insert into that hydrophobic environment of the membrane. The signal recognition particle is an important component of this machinery, and this happens on the surface of the rough ER membrane. So the signal recognition particle directs the polypeptide that is emerging out of the ribosome to a receptor that then initiates a translocation process. So this means there are pores of this protein translocated that allow big bulky parts of the protein to go through. We only talked about the sugar addition, right? But another thing that happens in the lumen of the ER is disulfide bond formation that helps proteins to fold the right way. Right? This means so if you have disulfide bonds, it's going to hold folds of the protein, and this happens also in the lumen of the ER in conjunction with glycosylation. And the recognition of the glycosylation is one of the mechanisms, right? Of course, in cells, there's always layers and layers of complexity. What is important is to understand the conceptual, that the cell really developed mechanisms of using tags. And end-linked glycosylation is one of these tags that tells the cell that this protein is folded properly. And then it's released to get out. If they don't fold properly, then they get the bigoted tags, and they are degraded after they are out of the unit. Right? So these are the major conceptual things that cells have to do, and of course there's many, many labs still working on the details of this. These are very complex machineries doing this. So conceptually, right, if we think about this, we've been talking up to now about this sequence that proteins have that determines the import into the ER, right? Because it's on the outside, the cytosolic side where the ribosome is. So this is just one example. So what is typical of these import signals is that it's something like 10 hydrophobic amino acids. So a sequence of hydrophobic amino acids. And like I showed you before, right, when we do these hydropathy plots, usually this is one of the first things that you could do looking at sequences. If you see a sequence of hydrophobic amino acids like this, you know that this is probably a transmembrane protein that started outside in the membrane of the ER and maybe ends up in the plasma membrane or in lysosomes. Then we're going to look at this again, but what is really neat is that there's a second signal. It is usually on the C terminus of proteins that does the opposite jobs, that keeps proteins in the ER. I gave you several examples of enzymes that have to be in the ER because they are there to glycosylate and deglycosylate and recognize folding. So these proteins have what we call the KBL sequence, which is this sequence of 4 amino acids that are recognized in the ER as a retention signal. So when they start getting out, they're brought back. So these are the signals that work in the ER. We're going to talk later when we talk about mitochondria into the really interesting different systems that evolved. And interestingly, right, since mitochondria and plastids had a bacterial origin, these signals are different than the signals that we see in the ER. And I'll just talk to you a little bit about the nuclear import, because this is pretty different, right? So this means we were talking up to now about signals that really take a protein that is on the cytosolic side being synthesized and across a hydrophobic bilayer into the lumen of the ER. So proteins also have to go in and out of the nucleus. But the way they do this is very different, right? So when we talk about nuclear import or nuclear export, we are not talking about translocation across the membrane. What are we talking about? So how does nuclear import work? It's not across the membrane. The pore, right? So everybody heard of nuclear pores? So what is really interesting about the ER is, so cells also have to solve this problem of how to send proteins in, right? This means you have to organize the chromatin. There's the whole machinery for transcription that happens inside the nucleus. So this is also still in the works, but the really important concept here is that the nuclear membrane, that we always draw it separately and put a little circle around the nucleus. Actually, what the nuclear membrane is a continuation of the ER. Right? So the nuclear membrane is formed of these flat cisterna that are completely continuous with this network of the ER. But when it gets close to the chromatin, really surrounding the DNA, there are structural changes that are observed in this membrane, and the important one are the pores that appear. So there are these regular pores that are on the membrane, and then there are all these fibrils that also help control what goes in, because it has to be a controlled process. It's very important that all the proteins in the cytoplasm just don't start clogging the nucleus, right? So let's. This is a nice example with a fluorescent protein that people now know the signals of nuclear imports. So you can put the signals and control them in different ways and make proteins go in or out of the nucleus.
Nuclear import and export can be directly visualized in living cells that express the green fluorescent protein GFP fused to the gene regulatory protein NFAT. NFAT is normally localized in the cytosol and excluded from the nucleus. But when the cytosolic calcium concentration is raised, NFAT migrates to the nucleus. This is done here experimentally by adding an ionophore that allows calcium to enter the cells from the medium. Upon removal of the ionophore, calcium levels return to normal, and NFAT is exported from the nucleus. Re-addition of the ionophore triggers re-import of NFAT.
So this is something that it's really very easy to demonstrate, that people have shown this, that you can even attach nuclear import signals to inert particles like gold, and these very large particles are inside the nucleus. It's really fascinating. And the structure of this is it starts to get asymmetrical only when you think about the third layer proteins. So this is an interesting complex in which you have first what we call the membrane ring complex that defines the pore. Then there's a second layer, which are the nucleoporins, which form the inner pore. And then these nucleoporins have these filaments, which kind of form this mesh inside the pore that is really thought to work in slowing down the protein traffic. And then on the inside, there's this what people call the nuclear basket that are also projections out of these nucleoporins. And on the cytosolic side, there are these fibrils that seem to play a role in capturing the proteins that are supposed to be translocated. The way this looks on, so this is a different form of microscopy, but does everybody know how scanning electron microscopy works? Somebody nodded here, would you define for us? What's the name? Suction, sorry. So how does scanning work? Exactly. So this is the concept here, right? And this means that there's no sectioning going on. So what was done here, that they basically ripped off the plasma membrane and exposed the nucleus. And all you do is that come with a coating. Usually they use gold for this. So this means some heavy metal that is going to slow down the electrons. And then they took a picture of how the surface of the nucleus looks like. So these nucleoporins actually, so that you see these 8 arms. So there seems to be a helical structure that is defined by these nucleoporins, and usually the lumen always appears occluded, but this is kind of an artifact, right? Because you have to coat the whole thing with gold to see it. But you see these very regular structures on the surface of the nuclear membrane, and then if you go back to the old transmission EM and you cut this, right? So cutting across this membrane, you see here the regions of the pores that really show how the bilayer, right? This looks like ER, right? It looks like a cisterna, and this means each one of these is a bilayer, and then the pore really goes through these 2 bilayers on each side of the cisterna, and you can see again material inside that probably was just caught there. So what is fascinating about the nuclear transport is that there's a very elaborate and clever mechanism to really prevent proteins to go the wrong way, right? So this means that this is controlled by this GTPase RAN. So the bottom line is that cells have mechanisms to keep the inside of the nucleus, the area inside of the nucleus. RAN is in its GTP form. And outside of the nucleus, it's in the GD. And then another important component of this system is the nuclear import receptor. So there's a unique, it's not only one, there are several types, that capture the protein that has the signal, right? The protein has to have a signal that it's supposed to go into the nucleus. It binds to this import receptor, and then this protein starts moving down the pore. So when it's in the presence of RAN GDP, it keeps going, and then when it reaches the other side of the pore, and it's in the presence of RAN GTP, GTP competes with the binding of the protein. So GTP basically jumps into your nuclear import receptor, displacing the protein. So it's basically, it arrives there, and then there's RAN GTP waiting to displace the protein. So the protein stays in the nucleus, and this diffuses out, and then when it's hydrolyzed, the GTP regenerates the GTP, and the cycle starts again. So the important thing to remember here is that it's the asymmetry in the distribution of RAN GTP and RAN GDP that controls the traffic into the nucleus. And what is really cool is that the other way around, when proteins come out of the nucleus, this happens with different receptors, what we call the nuclear export receptors. But it works exactly the same way, but the other way around. So this type of receptor, the protein is, imagine inside the binding of the protein to the receptor is allowed in the presence of RAN-GTP. And then when this RAN-GTP plus proteins start moving out, when they get to the area in which there's RAN-GDP, the protein is kicked out of the receptor. So it just works the other way around with different receptors, of course, because they have to react very differently to the binding of RAN-GTP. And this is how the traffic is controlled in and out. Okay, so another fascinating thing with the nucleus, which is amazing that the cells do this all the time in every cell cycle, right? It's something we don't think about very often. But this nuclear structure that is this extension of the yard, it has all these elaborate pores and all this elaborate machinery for import. It falls apart every time that a cell condenses its chromosomes and gets ready to divide. So this is what is indicated here, right? So this means one important component of this nuclear envelope that we call are the lamins, these structural proteins that are on the inner side of these cisterna that form the nuclear membrane. And these lamins get phosphorylated. So this is a big part of the control of the cell cycle, right? There are cyclins, kinases that phosphorylate these lamins, and this makes them just fall out of place. And this basically, with the concerted action of a lot of other proteins, including these import receptors, this breaks apart the membrane. And it is the dephosphorylation that happens also kind of as the chromosomes are beginning to decondense and the cell is getting ready to get out of mitosis, that then the nuclear membrane reassembles. So it's a catastrophic collapse that is tightly controlled by a series of phosphorylations and other proteins that modulate the shape of these mechanisms. So just summarizing, right, so this means the important concepts here is that don't forget that the nuclear envelope is continuous within the plasmic reticulum, so it's not a simple bilayer structure. It's basically a cisterna with space in the middle. And several molecules, including RNA that is made in the nucleus, subunits of ribosomal complexes, and many other molecules have transported to the cytosol, and they usually have these ribonuclear protein chaperones that really complex with RNA to bring it to the cytosol, and they have to be imported through these spores, right, through this mechanism that we discussed, which are these long, complex pores that span the whole cisterna of the membrane. The diffusion is passive, but it is the gradient of these regulatory proteins like RAN-GTP that controls the direction of the transport. And import receptors recognize signals on the amino acid sequence, so that's why you have different signals for import and export, because you have different receptors controlling this. And GTPase RAN is really the main player here that controls the traffic. Okay, so what I would like to do in the last 15 minutes, does everybody have a piece of paper? If not, I have a blog. Just one simple thing. It's going to be worth 7 points. If everybody is written there. So just describe something you learned today and why it's important. Okay? Anybody needs paper right now?
That's true. It's just the big ones and the small.
Okay.